In this video, we will discuss about some of the plant-based lectin and its interaction with the cancerous cell in a way that it will trigger apoptosis in the cancerous cells. In this video, we will not cover some of the specific lectins that will trigger apoptosis. However, we will cover the pathways that might leads to apoptosis whenever the lectins interacts with the tumor cells. So there are different ways by which lectins can trigger apoptosis. Some of these ways are called some of these pathways are called intrinsic apoptotic pathways, and some of these pathways and some of the other ways by which lectins trigger apoptosis are known as extrinsic apoptotic pathways. So in the case of intrinsic apoptosis pathways, is that whenever it can happen in two different ways. First one is that lectin interacts with its cell surface receptor that are present on the cell membrane, and then it will trigger the downstream signaling pathways in the mitochondria. And the other way that it can act is through this endocytosis. Lectins will internalize within the cytosol of the membrane, and then it will enter into this mitochondria organelle. And through both of these means, the mitochondria will release cytochrome C, and at the same time it will trigger this ROS, that is reactive oxygen species. And this reactive oxygen species will regulate this BUX and BCL2 production, and simultaneously it will trigger downstream signaling pathways that will lead to the intrinsic apoptosis. And we can see that the P53 will also get the function of P53 will also get disrupted because of this DNA damage response. And this the P53 functions get disrupted in a way that it will not be able to prevent the apoptosis. However, what it will does is it will prevent the cells. It will leads to this DNA damage response and cell cycle arrest. That is, it will work towards repairing this DNA damage within the tumor cells, and that might ultimately leads to the cell cycle arrest. So this interaction of these lectins not only trigger apoptosis, however, it will also lead to the cell cycle arrest. In other way, it will provide a restrictions towards the uncontrolled cell growth rate of the tumor cells. So this is one of the way by which these lectins might trigger the intrinsic apoptosis pathway. The other way that it can triggers is that through these extrinsic apoptosis pathways. In this case, the lectins, when interact with the cell surface membrane, it will lead to the downstream signaling pathways, this P13K, and at the same time it will also upregulate this RAS signaling pathways and this NF-K beta signaling pathways, and both of these will upregulate this caspase 8-9 and caspase 6-7. And this caspase 8-9-6-7 will further trigger the downstream signaling pathways that will lead to this apoptosis. And these pathways are known as extrinsic apoptosis pathways, and they are triggered by the lectin interaction with the cell surface membrane directly. However, in this case, what happens when lectins interact with the cell surface membrane, it will lead to the downstream signaling pathways in the mitochondria, and that will trigger the apoptosis. So this pathways is commonly known as the intrinsic apoptosis pathways, and this one is known as extrinsic apoptosis pathways. And the one more function that we have left to discuss is about the cytochrome C. So what cytochrome C does whenever it comes into the cytosol of the cells, it will trigger the extrinsic apoptosis pathways through this APAF1 or this APAF1 and caspase 9 interactions. It helps in the complex formation of this APAF1 and caspase 9 that we can see here. With the help of cytochrome C, it is able to form a complex, and that will that will helps in this extrinsic apoptosis of the tumor cells. So this is so by finding out this kind of knowledge, there is a good possibilities that lectin can be used as a biomarker to trigger the apoptosis within the cancerous cells. And if you need to understand more about this plant lectin interactions with the cancerous cells and how they will trigger apoptosis in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.